Hello and welcome to Nikolai's Genetics Lessons, and today I got interesting problem from one of my subscriber from Kazakhstan, and that person told me that this is high school level problem, but actually not probably all students of the college would be able to solve it, but those people who watch my videos regularly would be able to solve such problem very easy. So here is the problem. In the fox, there are 9 code colors: red, standard silver, Alaskan silver, double black, smoky red, cross red, blended cross, substandard silver, and sub-Alaskan silver. A red fox were crossed with a double black fox, and their offspring were then crossed with each other. The F2 phenotypes were 10 red, 18 smoky red, 20 cross red, 39 blended cross, 9 standard silver, 19 substandard silver, 12 Alaskan silver, 22 sub-Alaskan silver, and the last one 8 double black. And here's the answers to choose from. And I also forget to mention that this is not standard problem for standard high school level. This problem for Olympic games in biology. So Olympic games can be not only in sports, but also Olympic games are held in different sciences for high school students and college students. So what would be our first step? Of course, we have to convert these absolute numbers into ratios. For example, what minimum number would be here? 8, 12, 9, 10. So this is going to be our minimum that we would use 1 instead of these numbers. So 10 red would equal to 1, and 18 would equal to 2. So 10 is about twice smaller than 18, or 18 about twice bigger than 10. And these numbers shouldn't be exactly like twice bigger or twice smaller, because this is genetics and we are talking here about probabilities. So we are not looking for exact numbers like 10 and 20. And this can be just approximation. For example, here we may have 9 and here we may have 19 or 20. So next we have 20, that is cross red. So this is once again ratio is 1 to 2 to 2. Then we have 39, and this gives us 4, and then 9 standard silver, which gives us ratio to 1, and now we have 19 substandard silver, which gives us ratio 2, and 12 would give us once again ratio 1, and 22 would give us 2, and the last one, 8 double black, would give us 1. So this is our ratio, and actually those people who watch my videos regularly can right away see a solution here, and this is how we are going to solve. We have to add all these numbers, and if we add all these numbers, we are going to get 16. And this number would tell us right away that this trait under the control of 2 genes and 4 alleles. By the way, the funny thing that there is no such answer here. So sometimes I would say that about maybe 1% of all the problems in genetics have mistakes, whether in the problem itself or in the answers. So this is common thing. So instead of putting correct answer here, I want to show you how to solve this problem, and I'm going to explain what would be the correct answer. So now, with the next step, what we can do, we can rearrange these numbers, so we would have something more standard, like 1 to 1 to 2 and to 2 to 4 to 2 to 2 and to 1 and to 1. So as you see, here we have all the same numbers, but these numbers now rearranged. So we have 1 here, 1 here, 1 here, and 1 here. Here we also have 4 ones. Here we have 4 twos and 4 twos here, and number 4 is only seen here once and only seen here once. Why I know instantly that this trait is under control of 2 genes with 2 allelic forms, because when we add these numbers we get 16. That give me a strong hint that here we have dihybrid, standard dihybrid cross. And we were told that we cross one genotype that was, let me see, red fox, so red, and I have a suggestion that genotype would be capital A, and capital B, and we cross with another phenotype that is double black fox, so genotype can be small a, and small b, and this is parental generation, so this is parent 1 and this is parent 2, and in F1 generation we would have all the progeny of one phenotype and of course one genotype, capital A small a and capital B small b. And according to our problem, once again, red fox were crossed with double black fox, and their offspring were then crossed with each other. So this is the offspring, and this offspring were crossed with each other. Because all the offspring of the F1 generation has the same genotype, that means that we have to cross with the same genotype. So F1 among each other. And as you see, this is exactly what we call dihybrid cross. So we have gene A and gene B. So once again, only 2 genes here, and each gene represented here by 2 alleles. So this is classical dihybrid cross, because D stands for 2 genes, and hybrid stands that each gene is in heterozygous form. And what we are going to get in the next F2 generation, once again, this is classical dihybrid cross. So each parent here in F1 generation would produce following gametes. The first variant would be capital A and capital B. For example, this parent may give to the progeny these 2 alleles. Once again, now we talking about gametes. Gametes are haploid, and this is first variant. Second variant, this can be dominant allele A and recessive allele B in the gametes. So capital A and small b. Another variant of the gamete can be recessive allele A and dominant allele B. So recessive allele A and dominant allele B. And the last variant would be recessive allele A and recessive allele B. Recessive allele A and recessive allele B in the gametes. Because both parents has the same genotype, both parents also would produce the same type of the gametes. So here on the side I'm going to put the same 4 variants of the gametes: capital A, capital B, capital A and small b, and small a and capital B, and the last variant would be small a and small b. Now we build a Punnett square. As you see, we are going to have 16 cells. Do you remember number 16? That gave me right away a hint that this trait under the control of 2 genes, and each gene represented by 2 alleles. So now let's take a look what we are going to have, for example, in the first cell. Capital A and capital B here. Once again, this is gametes of one parent, and of course 2 parents have to be of the different sex. So we may say that this is sperm, and another parent would contribute gametes in the form of egg cells. So for example, this is going to be male side, and here we are going to have female side. Because both parents, once again, of the same genotype, we would see the same variants of the gametes. Now let's find all the variants of the genotypes in their progeny. And by the way, this is going to be F2 generation. So capital A and capital B, small b here, capital A, small a, capital B here, capital A, small a, and capital B, small b here, and capital A, capital B, small b, capital A, small b, and capital A, small a, capital B, small b, and capital A, and small b here. I will speed up my video. So I finished my table, and now let's find out if this table would be able to give us results that we got earlier. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9 different phenotypes. So let's take a look at our table, and you would see, for example, that this genotype, which equal to phenotype, we can see in our table only once, and this genotype and phenotype we can see only once, and this one, and of course this one too. And by the way, these two, as you remember, were our parental genotypes. So one of these would produce red color in fox, another one black color in fox. And once again, by this diagonal, we would see that these phenotypes only would repeat once. So this is going to be this one, this one, this one, and this is going to be what color? Red, pink, white and yellow. So I'm missing red here. So as you see now, we have 4 different phenotypes and genotypes, and here in this table genotype equal to phenotype. So Now let's count how many genotypes repeated here twice. For example, this genotype here is repeated twice. So let's underline this too. And next, genotype and genotype equals to phenotype. We only can find in our table twice. So let's underline this too. Next is different. It is repeated 4 times, and this is going to be this genotype. So it is heterozygous for both genes, capital A small a and capital B small b. As you see, these genotypes forms another diagonal. So all these genotypes are the same. And we see that this genotype would repeat 4 times. So by this diagonal we see genotypes that would repeat only once, would be unique. By this diagonal we see that genotype would be the same and would repeat 4 times. So this is this 4 in our ratio. Now let's go over this two, and this is going to be a genotype that is next one. So let me choose another color. So this is going to be this one, capital A and small a, small b, small b. And we see this genotype here. So once again, it is repeat twice. So we underline this 2. And the last one also have to be repeat twice, and this is going to be this genotype here. 1 and second. As you see, the same genotype, small a, capital B, small a, and capital B, small b. So once again, we have total number, if we add all the phenotypes, which equals 16. And if our table here 4 by 4, we have 16 cells here, and we have, as you see, 9 different unique phenotypes, and this is exactly what we see here: 1, 2, 3, 4, 5, 6, 7, 8, 9. And as you see, we have the same balance between all these different genotypes. And genotype here equal to phenotype, we would have the same ratio as in our problem: 1 to 1, 2 to 2 to 4 to 2 to 2 to 1 to 1. So as you see, this trait, which is color, under the control of 2 genes. Once again, this is not 4 genes. Here is a gene A, here is a gene B. And each gene represented by 2 alleles. It can be with a recessive allele, or it can be dominant allele. Once again, here we see that this parent is heterozygous for both genes. And of course, as you see, there is no such an answer here. The correct answer would be that this trait under the control of 2 genes, and each gene represented by 2 alleles. But answer B is not the correct one, because 4 alleles is not the same as 4 genes. Alleles are just slightly different forms of the same gene. And once again, here we have 2 genes, and each gene represented by 2 alleles, which is slightly different forms of the same gene. So the correct answer would be that this trait under the control of 2 genes that is represented by two alleles. And let's say that this is answer F. And instead of genes, we also can use as variant of the answer two loci, and each locus have two variants of alleles. And this answer is not the same as answer B, because 4 pairs means 8. And 4 genes or pairs of genes, of course, would mean 4 loci, because each gene occupies a unique place or locus on the chromosome. So as you see, the correct answer is answer F. And one more comment would be, when you would see such a complex problem, start with finding ratios. For example, in classical dihybrid cross from your textbooks, you probably remember ratio 9 to 3 to 3 and to 1. If we add all these numbers, we are going to get 16. Within last month, I also show you different videos with also other ratios that also would give 16 if we add all the phenotypes. For example, 12 to 3 to 1 also would give us 16. So this is going to be a strong hint how to solve such problem. And I also forgot to mention why, for example, all these genotypes is not equal to one phenotype. For example, here we have genotype that is homozygous for the gene A and homozygous for the gene B. And for example, this genotype in classical Mendelian genetics also would mean the same phenotype. But here we do not have classical simple dominance, but here we have example of the additive trait. So each unique genotype also would produce unique phenotype. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.